So in 7.33, we're doing the next part of the now solve this question for this chapter, and they divided it into, they put the second part in the problem section of the chapter. So problem 7.33, when the cloned cat carbon copy was born, she had black patches and white patches, but completely lacked any orange patches. The knowledgeable students of genetics were not surprised at this outcome, even though I was a little surprised, but of course we should be surprised because the scientists didn't even know what to expect. So that is not true. It was okay that I was surprised. So starting with the somatic ovarian cell used as the source of the nucleus in the cloning process, explain how this outcome occurred. So it appears, now you should go back and take a look at that now solve this problem if you haven't, so you can understand what we're talking about here. So it appears that Rainbow, a calico cat with the genotype CB-CR, CB producing the black pigment, CR producing the red pigment, we took a somatic ovarian cell. We know it's somatic now because the problem tells us. And that somatic cell is going to have an X chromosome in the Barr body. And we're going to say the X chromosome with the CR allele. And here, this is a nucleus here from that ovarian somatic cell, and the CB allele is on the active X chromosome. This was put into the enucleated egg. So here it is, and then this was the one that was put into the surrogate mom. Surrogate mom. Okay, thank you, surrogate mom, for giving birth to the first cloned cat, which is carbon copy. So, and carbon copy has black patches and white patches, but no orange patches. So, scientists have learned something, at least with this one experiment, with this one cloned cat. And I don't know how many cloned cats have been cloned since carbon copy. And I'm not sure how carbon copy is doing. I think carbon copy actually did quite well. At least I hope so. But what scientists now know is that, you know, when this what we can think from this one experiment is that the CR allele was in the Barr body of the enucleated of the nucleus from Rainbow, and when we put that nucleus into the enucleated egg, it remained in the inactive state in every cell that resulted from mitosis. That descended from I should say not from mitosis, because all of the cells are going to be created from this one, but so this was the zygote, and this is the one that forms the embryo and makes thousands of cells, and in each of those cells the CR X chromosome, the X chromosome with the CR allele, remains inactive. So that appears to be what happened. And I guess the another point to make is that at no point during this process did both alleles, it appears that at no point during this process both X chromosomes were never active in the same cell, because if that were the case, then we would pursue the normal path of development where cells develop for a while with 2 active X chromosomes, and then at some point 1 to 3 weeks after during embryogenesis, after about 3 weeks, at least in humans, I'm not sure how long in cats, then the X chromosomes are inactivated randomly. That doesn't seem to have happened. It seemed that carbon copy, when it started as this one cell, one nucleus in an enucleated egg, 1 X chromosome was inactivated, and this same X chromosome remained inactivated in every cell during development. So something interesting about cloning organisms, something scientists could not have predicted. And again, we still don't know much about X chromosome inactivation and how that whole process works. We know little bits, but it's a very fascinating subject. Okay, so I hope that helps answer that question, and you can come see me if you have some additional questions on 7.33 or any problem in Chapter 7.